Hello friends, in this video we are going to discuss about transfer RNA, full form transfer ribonucleic acid. First of all, Francis Crick postulated the presence of adapter molecule and it was seen that it performs two functions. Read the code that is present in messenger RNA, second bind to specific amino acid. Later on, this molecule was referred as transfer RNA. This transfer RNA molecule acts as intermediate molecule between triplet code of messenger RNA and amino acid sequence of polypeptide chain, that is protein, which is synthesized during translation. Now the occurrence of transfer RNA. Transfer RNA occurs in cytoplasm of a cell. Transfer RNA molecule is much smaller than ribosomal RNA molecule. There are over 60 types of transfer RNAs and all of them have almost same basic structure. These molecules are 70 to 90 nucleotides long. If we talk about its secondary structure or 2D structure, it is like a clover leaf, and its tertiary structure or 3D structure is inverted L shape. This structure was proposed by Kim and Klug, and this is the actual structure of transfer RNA molecule. If we see the secondary and the tertiary structure, both are folded structure which are formed due to hydrogen bonding between complementary bases. This is the two-dimensional structure of transfer RNA, which is clover leaf structure. Let us see the structure in detail. Clover leaf structure, it is secondary folded structure. It has 3 hairpin loops, giving it an appearance of 3-leaved clover. Now, main parts of this clover leaf structure. First is acceptor arm, which is formed by the base pairing of 7 to 9 nucleotides of 5' and 3' terminal. 5' terminal has a phosphate group. 3' end has a specific sequence of CCA tail. Amino acid attaches to 3' hydroxyl of the acceptor arm. So this is the acceptor arm. It is formed by the base pairing of 7 to 9 nucleotides of this 5' terminal and 3' terminal. 5' terminal has phosphate group and 3' end or terminal has specific sequence of CCA, or we can say it has CCA tail. To this 3' hydroxyl group of acceptor arm, specific amino acid attaches. So it is known as amino acid attaching site. Now second is DHU loop. D arm has a stem of 3 to 4 base pairs and it ends in a loop called D loop. This D loop contains dihydrouridine, which is a modified nucleotide. This DHU loop or D loop is also known as amino acyl synthetase binding loop. So this is the DHU loop or D loop. It has a stem which has 3 to 4 base pairs and it ends in a loop known as D loop. It contains dihydrouridine, which is modified nucleotide. This loop is also known as amino acyl synthetase binding loop, because to this loop attaches the enzyme amino acyl synthetase, which is involved in the protein synthesis or translation process. Third is anticodon loop. It has 5 base pair long stem. It has an anticodon loop which contains complementary codon, which is 3 nucleotide sequences, and this is complementary to the codon present on messenger RNA. And for this messenger RNA codon, transfer RNA carries amino acids. The unpaired bases of anticodon loop pair with messenger RNA codon, and this codon is identified by specific transfer RNA. So this is the anticodon loop which has 5 base pair long stem, which then ends in a loop. This loop has 3 nucleotide sequence codon, which is complementary to the codon present on the messenger RNA. So this anticodon, which is unpaired, base pairs with codon present in the messenger RNA, and this messenger RNA codon is identified by specific transfer RNA. Now next is tRNA loop. The T arm consists of stem of 4 to 5 base pair and a loop containing pseudo-uridine, which is modified uridine. It is ribosomal binding loop. So this is tRNA loop, which has stem which contains 4 to 5 base pair, and it also has a loop which consists of pseudo-uridine. This pseudo-uridine is modified form of uridine. This is ribosomal binding loop, which binds with ribosome during protein synthesis. Then fifth is variable loop. This is present between the tRNA loop and anticodon loop. Its size varies from 3 to 21 bases. It helps in the recognition of transfer RNA molecule. So this is the extra arm or the variable loop. This is present between anticodon loop and tRNA loop. Its size varies from 3 to 21 bases, and this loop helps in the recognition or in identification of transfer RNA molecule. Functions of transfer RNA. It plays an important role in protein synthesis. It acts as an adapter molecule for linking amino acid to its specific codon present in messenger RNA. It transfers the amino acid to the growing polypeptide chain in the ribosomes. It decodes codons of messenger RNA. It translates entire sequence for a polypeptide chain. Now, amino acid activation and involvement of transfer RNAs. How? Specific amino acid, depending upon the codons on messenger RNA, attaches to specific transfer RNA, and then it is attached to the growing polypeptide chain during protein synthesis. Attachment of specific amino acid at the 3' end of specific transfer RNA process is known as amino acid activation, and it is catalyzed by amino acyl transfer RNA synthetase. The reaction of this process is amino acid plus ATP. ATP is used as the source of energy. In the presence of enzyme and Mg2+ gives amino acid AMP enzyme complex, and inorganic phosphate is released. Then amino acid AMP enzyme complex plus transfer RNA gives amino acid transfer RNA. AMP and enzyme is released. So the transfer RNA helps in the attachment of amino acid to the growing polypeptide chain during protein synthesis. In this way, translation or protein synthesis takes place. So this is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.